So as a continuation of the last video, in this video I will touch on the topic of acid-fast staining. And for those of you who haven't watched the previous video, well, in that video I talked about the basics of staining, and also I talked in detail about gram staining. So if you watch that video before watching this, I think it will be at least a little bit easier for you to understand whatever I explain in this one. So to review a little bit, there are two main types of staining. We have the gram stain and the acid-fast stain. Well, as most of the bacteria take the gram stain, obviously, it's the most important stain we will know in microbiology. But in today's topic, the focus will be on acid-fast stain or Ziehl-Neelsen stain. Although only a few bacteria and fungi and some protozoans take up the stain, it's important we know about acid-fast in detail, because essentially Mycobacterium is the only genus of bacteria that take up the acid-fast stain. So Mycobacterium tuberculosis is the pathologic agent of tuberculosis, and it's obviously a really prevalent disease, especially in the developing and also underdeveloped world. So if we know about acid-fast stain in detail, it would be very easy for us to make the diagnosis in the lab about Mycobacterium, and we can help the clinicians, or if we are the clinicians ourselves, we can start the therapy as fast as possible. So here I have included two pictures from the last video. These are gram positives and these are gram negatives. And about this picture, I will discuss as we go on with the video. So why not use gram stain also in case of acid-fast species like Mycobacterium? So the main difference is in the structure of the cell walls. Here we see a new component. We see mycolic acid. So this mycolic acid is the component due to which we cannot use gram stain when we are trying to stain the acid-fast organisms. Well, what's different about mycolic acid? It is actually lipid soluble, because mycolic acid is actually a lipid compound. So as we know from basic chemistry that like dissolves like, right? So only polar solvents are able to dissolve polar compounds, and non-polar solvent will be able to dissolve non-polar compounds. So mycolic acid would be a lipid soluble. That means a non-polar solute or non-polar compound which will dissolve in a non-polar stain. That's why we cannot use gram stain to stain anything containing mycolic acid. And in case of mycolic acid containing acid-fast organisms, the primary stain we will use will be carbolfuchsin. So there are a few terms that I mentioned in the past video. Just to review a little bit, primary stain will be the stain that we will use at the very beginning, and trapping agent will be the agent or will be the compound that we will use to trap the primary stain inside the cell wall. Whether it's an acid-fast or a non-acid-fast organism, it doesn't matter. Primary stain will colorize both the type of bacteria or organisms. So a few examples here. Carbolfuchsin is the primary stain we use in case of acid-fast, and heat is actually the mordant or trapping agent that is used in acid-fast staining. And decolorizer, well, first we said that primary stain has to colorize both the type of bacteria in the same way. So how do we know which ones are acid-fast and which ones are not? That's why we use a decolorizer to differentiate between the types of bacteria. And after we have successfully decolorized one of the types of bacteria, to confirm or to visualize the colorless bacteria, we use a counter stain. And again, for a few examples, acid alcohol would be our decolorizer of choice. In case of acid-fast stain, and actually it's a mixture of 3% hydrochloric acid and 95% alcohol. And our counter stain will be methylene blue. So moving on, now let's talk about why is it that we use the term acid-fast when we talk about the Ziehl-Neelsen stain. Well, we will get the answer as I describe the process. Before that, let's clarify that the circular ones are the non-acid-fast bacteria, and the rod-shaped ones will be the acid-fast ones. So firstly, we use primary stain carbylfuchsin to colorize both of the type of bacteria. Then we heat up that slide to trap that primary stain carbylfuchsin inside the cell wall of the acid-fast bacilli. And as a result, what happens is the acid-fast bacilli retain the primary stain carbylfuchsin, but the non-acid fast ones are decolorized. Well, why is it that the acid fast bacteria are not decolorized at this step? Because the mycolic acid has a stronger affinity towards the primary stain carbylfuchsin than it has affinity for the acid alcohol. So that means the acid alcohol will not be able to take the primary stain out of the acid-fast bacterial cell wall. So it will still retain the primary stain. And at the last step, to visualize the decolorized non-acid-fast bacteria, we use methylene blue, and we see that the non-acid-fast bacteria are stained at this step. So in the end, what we get is red colored acid-fast bacilli and non-acid-fast purple colored bacteria. So that's the counterstaining step. And now if we look at the actual slide, well, the blue ones will obviously be the ones which took up the counterstain at the last step, and the red ones will be the ones which have retained the primary stain from the first step. So the red ones will be the acid-fast bacilli, and the blue ones will be the non-acid-fast bacteria, or any other species for that matter. Now, if we look at a few examples, well, first, the very classic picture of red bacilli against a blue background. Well, why is it that way, that Mycobacterium tuberculosis stain this way or look this way? Well, because the red color is due to the primary carbylfuchsin stain and the blue background is due to the methylene blue counterstain that we used. And that's why we get this red bacteria against a blue background classic picture. Also to mention that Mycobacterium tuberculosis are also commonly known as acid-fast bacilli, or AFB in short. So that's something you will see being mentioned in your books over and over again. Now, to broaden our knowledge about acid-fast stain a little bit more, we should also know that Nocardia is another bacteria that also take up the acid-fast stain. And if we want to be technically more correct, then we can say that they are partially acid-fast. Why? Because few segments of the bacteria take up the methylene blue stain. So what happens is they're not fully acid-fast. They're not fully red. Some parts of them are blue and some parts of them are red. And if you ask why is it that these bacteria look like fungal hyphae, because Nocardia along with Actinomyces, if you remember, these two are actually branching rods. They're not like usual bacteria. They're not cocci or bacilli. They're actually branching rods. That's why they look like fungal hyphae. Now, a few other examples. It is nice to know that Histoplasma is a fungus that also takes up acid-fast stain. And also importantly, Cryptosporidia and Isospora are two protozoans, and the eggs of these species take up the acid-fast stain. And in this picture, I have tried to demonstrate the Cryptosporidia eggs. So these circular ones are the eggs of Cryptosporidia, which have taken the carbylfuchsin stain and has retained it. So these look red, and this is our classic blue background. So this is also an acid-fast staining example. So now, to summarize the acid-fast stain, I've included this chart. Well, it's the same thing actually, like the procedure. So firstly, we use a primary stain, carbolfuchsin, and both the acid-fast and non-acid-fast are stained red. So in the next step, the decolorizer is used, and only the non-acid-fast bacteria are decolorized because of the absence of mycolic acid in their cell walls. And the acid-fast bacteria having the mycolic acid will retain the primary dye. And then we use the counter stain methylene blue to actually visualize the non-acid-fast bacteria that were colorless at the last step. So what we get in the end is, for acid-fast, a red colored species against a blue background. That's the classic picture that we should remember at all times. Now, acid-fast stain is something that's still used in the developing and underdeveloped world to visualize Mycobacterium tuberculosis, but for the developed world, they are using a different type of staining. They are using a fluorescence microscopy based stain, that is auramine-rhodamine stain. And what's special about it is it's fluorescent, right? So we see these bacilli against these fluorescent bacilli, to be more accurate, against a dark black background, and these would be Mycobacterium tuberculosis, and these are, as I demonstrated in the last slide, the eggs of Cryptosporidia, and both are fluorescent in these slides. And to talk about a few advantages of this type of staining, well, it's more sensitive than the Ziehl-Neelsen, and it takes less time than the acid-fast staining. And also, we don't have to use any oil immersion lens. But there are some disadvantages as well. That is, it's obviously more expensive because we have to use fluorescent microscope, and it's not something that every lab has. And also, it's not as specific, so it will stain many other things that are not acid-fast. So if we want to be certain about our diagnosis, we should go back and do the acid-fast stain after we have done the auramine-rhodamine stain. And also, it's important to notice that if the auramine-rhodamine stain comes up negative, it does not mean that we don't have acid-fast bacilli in our sample. Well, what we have to do then is, to be more sure, we have to go back and do the acid-fast stain again. So it's actually a mixed experience. There are advantages and then there are disadvantages, but actually in the developed world, as it takes up less time and they have the equipments to actually do the staining, so they opt for auramine-rhodamine stain before they do the acid-fast stain. That's actually it about acid-fast staining in this video. I think I've been able to explain rather clearly about what the steps are of acid-fast staining, and also the examples. So to review the absolute high yields of this video, first obviously we have to remember this classic slide of red bacteria against blue background of Mycobacterium tuberculosis. And also, it's good to remember the procedures that are associated with acid-fast stain. That is, we use carbolfuchsin as the primary stain, then we use the heat as mordant, and also an acid alcohol to decolorize. And as the acid-fast organisms retain the primary stain because of the mycolic acid content in their cell wall, they're still in the end red bacilli. So that concludes this video, and there are a few stains that I haven't mentioned in my first two videos. So those would be used to recognize or identify the species that do not take either the gram stain or the acid-fast stain. So there are some miscellaneous stains that we need to talk about, which I will in the next video. So till then, keep learning and stay sharp.